Okay, Sterile Insect Technique. Sterile Insect Technique is awesome. Basu, how many times have you been told the story? Have you ever had a lecture on Sterile Insect Technique yet?
No.
Never? Okay. I thought for sure...
I have heard about it.
But you've never had a lecture on it? Okay, I thought entomology people would have had this like 50 times. Have you had this before? So the guy who invented this is Nippling, and it came out in 1955. So we'll talk all about this and how it relates to medical entomology. So first we'll talk about the screwworm. This is for veterinary importance, and humans also have some minor myiasis issues, which we'll talk about. So myiasis is when a body gets infected with larvae, fly larvae, that eat your flesh. So that's myiasis, is like an infection of fly larvae. So the screwworm, the species is Cucleomyia hominivirax. Okay, this is the life cycle. You want to know this? You want to know what the screwworm species is because it's super famous. Okay, so basically the life cycle of a screwworm is a gravid female. So that means like a female that's mated, that's ready to lay some eggs, will find a cow or a vertebrate, okay, that has a wound on it, and then it will lay its eggs right by the wound. And then the eggs hatch, and the larvae literally eat the flesh of the animal, and then they fall off and they pupate, and then they become adults, they mate, and the cycle repeats itself. And the larvae will just keep getting laid on this cow because the wound will get worse and worse and worse, and they will literally eat the animal alive. So you get mortality from this, and it was a massive problem before we got rid of it. So one thing to note is the females deposit in the near the open wound, oviposit, and the larvae literally will just dig into the flesh and eat it.
Like they do in your food?
What?
Like they do into food, like in the lab?
Yeah. So here's what the larvae look like. They got these like hooks. Here's what the adult, this is actually a blowfly. So I also just put this slide in is a lot of these flies that cause myiasis, so these flies that will eat flesh, are important for forensic entomology. So this is just a little side note on forensic entomology, that because the flies lay in the flesh, if you have a dead body or if you have some corpse, oftentimes they'll use the larvae and the adults of these flies to judge how long the corpse has been there based on the different instars of the larvae. So the most common myiasis problem in like corpses are caliphorids. So those are blowflies. These are the metallic green flies that you see if you have like a dog and your dog craps outside. Usually these, you'll get blowflies. And these, I actually got contracted to, there was a lawsuit, and a daughter had come in to visit her mom in the nursing home, and they found like worms in like the mother's, like, I guess you'd call them skin folds. They found like worms in there, and they sued the nursing home, and then the nursing home basically needed to find out like how long the flies had been there without being cleaned. So unfortunately they did not have samples, but they had like a movie of these larvae. So they sent me the movie, and I had to like determine from the movie how long the flies had been there based on like judging the size of the larvae. So just a side note, if you ever think about forensic entomology, it relates to myiasis, and myiasis is when flies infest the human body. So there's the differences that you want to know. Primary myiasis, which is like screwworm, is organism feeds on living tissue. Okay, so there's some flies that can take tissue that's alive on a live animal and eat it alive. There's other flies that can only eat dead tissue. So if the organism is dead and it's rotting, there's some flies that can only eat the dead tissue. That's called secondary myiasis. So the screwworm is primary. It can eat you alive. So this is Nippling. We'll give you the basics of the sterile insect technique. So he invented this technique in 1955. And the idea is, if you go through this figure right here, you take a bunch of males and you can irradiate them with radiation to make them sterile. Okay? Then you release millions of sterile males, and the sterile males mate with wild females, and then the females lay eggs and they don't hatch. So you basically outcompete wild fertile males by releasing millions of sterile males. That's the basic strategy. So a key thing here is they have to outcompete the wild males. So you can't irradiate them and make them sick and just release a bunch of like sickly males. You have to irradiate them or sterilize them in a way that does not inhibit their fitness in any way. Does that make sense? So it's a key point. You can't hurt them. So he thought of this idea, but and I'll give this as a nod to Drosophila research, which I'm a big fan of. So Knipling didn't necessarily think of this idea. It had been done before in Drosophila in researchers who had been using irradiation to sterilize fruit flies for genetic experiments. And he was talking to those researchers who were working on fruit flies, and there was a conversation where he basically then that gave him the idea to try this in the screwworm. So a lot of people question what kind of positive, I guess, research comes out of working with fruit flies, Drosophila, but it's a basic model system of insects, and oftentimes things that work in other insects are first developed in Drosophila. So that's just a basic pointer there. Okay, so there's 4 things that are necessary for sterile insect technique, and I will ask this on the test, okay? You need a way to rear millions of insects. You need a way to exclude females. You need a way to sterilize males without reducing fitness. And you need a way to release the insects, okay? So all these things need to happen for sterile insect technique to work. So we'll talk about each of these points. And sterile insect technique, there's many different applications and many different types of how people are doing it. So there's a guy named Zhiyang who works at Michigan State, who works on Wolbachia. So he basically takes Wolbachia and puts them in insects, and then they rear millions of sterile mosquito males that are sterilized by the Wolbachia. So this is a picture of Zhiyang's factory in China, where they are rearing millions of mosquitoes. So this is getting big in China, where they're building these big factories. There are factories in Brazil that belong to a company called Oxitec, which was just bought by an agricultural company. So Oxitec was started in Britain, and they have a different strategy, but they basically are doing sterile insect technique with mosquitoes. So there's 2 kind of big competing strategies using sterile insect technique and mosquitoes. One uses Wolbachia, and another one uses what's called RIDL, which we'll talk about in the Oxitec strategy. And now there's factories in California, which are owned by Google, and Google owns a small life sciences company called Verily Life Sciences, which is doing sterile insect technique releases in California and now all over the United States. So there are factories here in America. And with the screwworm, there is a factory in Panama, which we'll talk about. So the sterile insect technique works so well. The screwworm used to be all throughout southern United States, okay? And we basically pushed it out of Florida up and then all the way down through South America down to Panama. So if you don't know geography of Central America, this is Panama, okay? And we've pushed the screwworm all the way down to Panama so that it's in South America, but it never comes up. And it never comes up because the USDA and the Panama government maintain a factory that looks like this in Panama, right here at the isthmus. And every week, they are rearing 20 million sterile screwworm pupae. They feed them on cow blood. This is what they look like. So these are factory workers rearing screwworm, taking out the pupae to be ready to be irradiated.
Interesting job.
Yeah. It's crazy when you find out all the kinds of weird jobs there are. These are the screwworm adults that they sterilize. And we'll get into a little bit how they sterilize them in a little bit, but I just wanted to show you that for the first point, the thing you need a factory to rear millions of insects. For the first point, the thing you need a factory to rear millions of insects. Okay, so they literally build like factories to rear these insects. Okay, you need a way to exclude females. So you can't rear a bunch of screwworms or mosquitoes and then try to do the sterile insect technique and also release females, because then you're just literally increasing the population, okay? You can only release males. So that means if you rear them, 50% of your reared insects are going to be female. So you have to have a way to selectively kill females. So you have to have a way to selectively kill females. Inside China's mosquito factory. This is Zhangji's factory. So look at what they have here. This is a screen, and the screen, when they take, these are larvae and pupae, and they can take these mosquitoes. It'll show you more. Okay, here's where they're micro-injecting them with Wolbachia. They don't actually do this in the factory. So here's the larvae bins. They're rearing the larvae. Sex separation area. So they're selecting out males versus females. So here what they do is there's this, can you see my mouse? Yeah. So there's this two glass panes, and they're separated by a tiny thickness, okay? And mosquito larvae, the female larvae are fatter than the males. Males are thinner. So all they do is they take millions of larvae and they run them through these two glass planes at a set thickness that stops the females and lets the males pass through. Okay, and this works with 99.9% accuracy. So here they're just pouring water with larvae, and look, there's the pupae. So these are the females, they get stuck. But the thin ones, the males, fall through. So then they have, that's how they get rid of the females, and then they release them.
That's when you take the little drop and run, right?
So only males are released. Males do not bite, so it works well. And they can reduce by 96% populations. So here they're driving around into little different neighborhoods, and then they'll release these. That guy's really happy. No more mosquitoes. That's it. Okay. So you need a way to exclude the females. Okay, so for mosquitoes it works well because there's this different size in the pupae. Not all insects are like that. So depending on the insect that you're targeting, you need to figure out a way to kill or get rid of the females. Okay, let's watch this video now. So this is Google's way to get rid of females.
What?
Okay, so Google has a special van that, like, they hold the mosquitoes and it pops them out as they drive around with this van. So they're targeting Aedes albopictus. So this is the way that they release them every week, a mobile unit. So they have this van, okay? And they load up their van full of mosquitoes. And in this van are little air chutes that shoot out mosquitoes as they're driving around the neighborhoods. So they're using Wolbachia to sterilize them. And they first trialed this in Fresno, California. Over 20 period weeks, so they're releasing continuously. Oh, I just missed that. Hang on. Is that it? There's one thing I wanted to show you. Where is it? Hang on. That's the one I want to show you. So they have a robot that will, as the mosquito, instead of doing it the mechanical way, to separate out the females, they have a robot that, as the mosquitoes come out of the pupal stage, a robot takes a picture of them with the camera, and through machine learning they can tell whether it's a male or a female, because there's a sexual dimorphism if you look at the antennae. And then if it's a male, they let it go out through a tube that collects males, and if it's a female, they let it go out in a tube that collects females. So they sex separate with this massive robot that can take pictures and tell whether it's a female or a male. And then they load the males into the van, and then they release them. Okay.
Is that 99% effective as well?
It's actually better. They gave a presentation at the Wolbachia meeting. They can reduce the error rate to like 1 in a billion. So they have 1 female per billion males is their contamination rate. And they're trying to get that even lower. So here I'll just say there's just a terminology distinction, which I think is stupid. And I don't use it. But if you see incompatible insect technique, that means they're using sterile insect technique, but it relies on Wolbachia. So because Wolbachia, we already had a lecture all on this. Wolbachia creates cytoplasmic incompatibility. So if you see IIT, that's using Wolbachia. If you see SIT, that's canonically like usually using genetic ways to sterilize or using radiation to sterilize. So that's just a little terminology difference. There's also a company in Kentucky that Verily collaborates with called Mosquito Mate, which was started by Steve Dobson at the University of Kentucky. So you need a way to sterilize males is a third one. There's different ways to do this. Let me get my laser pointer. So Nippling was old school, and he collaborated with the U.S. Army in Texas, and they basically took screwworms, and at first they were using x-rays. Later they got a radioactive cobalt source, and they would literally, here he's got a canister of pupae, and he pours them through this thing, and in this old tub in the basement somewhere in Texas is a radioactive source of cobalt, and it zaps them with radioactivity. They fall out and they're sterile. Okay, so they did it that way. Not all insects can be done this way because a lot of insects are not tolerant to radiation. So if you irradiate them, they get really sick. Okay, so it worked well because screwworms don't get sick from radiation. They basically just get sterilized. But you couldn't do this with mosquitoes because mosquito males get really sick when you treat them with radiation. So this strategy works with some insects and does not work with other insects depending on how sensitive they are to radioactivity. So there's different ways to sterilize. Okay? So there's irradiation, and if that doesn't work, you can try Wolbachia. Wolbachia is bacteria that secretes the SID-A, SID-B molecules. What we're working on is trying to build transgenic models of sterile insect techniques, so taking the genes from Wolbachia that sterilize and putting those directly into the insect. So that would be transgenic SID-A-B. RIDL, which is called release of a dominant lethal, which we're going to talk about extensively, which is the Oxitec strategy. There's chemical sterilization methods, and then there's methods where you can just manipulate the chromosomes and mutate certain genes to produce sterility. Okay, so these are all different means to sterilize the insect, and whatever insect you're working on might prefer one of these various methods. So let's talk about the Oxitec method. So Oxitec has been releasing now in South America for quite a while. There was a big story that before the Olympics in Rio, they contracted Oxitec to reduce the mosquito populations by using RIDL. So release of insects with a dominant lethal. This is a slightly different strategy. It's still a formulation of the sterile insect technique, but the technicality is that this is not a way that's sterilizing the sperm. What they're doing is figuring out a way to kill larvae. Okay, so this is a trick that kills the larvae. So what they do is they rely on this TET repression system. Okay, so first let's talk about the genetic regulation of how this works. Okay, so we know about promoters. We know how promoters regulate on and off status of a gene. So what they do is they take, so for RIDL, release of insects with a dominant lethal, they take a killing gene, let's say killing gene X. Okay, so if this gene is on, it'll kill the bug. Okay, and they shut it off. And they shut it off with a TET off promoter sequence that they insert right here. And then there's a second gene called TTA, which we'll talk about. So let's talk about the TET off system. So tetracycline is an antibiotic. In bacteria, there are genes that are regulated by tetracycline. So this promoter is actually a sequence from a bacteria. Okay? So this genetic system is from bacteria. And it's a method, the TET off system is a tetracycline-controlled transcriptional activation, is a method of inducing gene expression where transcription is reversibly turned on or off. So I said on or off. in the presence of tetracycline. Ignore that. Or its derivatives, doxycycline. So it's a way you can turn genes on or off by adding this chemical tetracycline. Okay, tetracycline works very well because it's pretty stable. So if you put it in a mosquito, if you make a mosquito eat it, it stays active in the mosquito for a while. Okay, so there's this protein called TTA. So this is the gene. The gene makes a protein TTA. And this is a fusion protein. That means this is not a normal protein you'd find in the wild. It's a chimeric. So they took two things and stuck them together. They took a sequence that binds tetracycline. Okay, so it binds the chemical, this chemical right here. It binds that chemical, and they fused it to a transcription factor. BP16. Okay, so this is a fusion protein. And in the TET-off system, you add tetracycline, it binds a transcription factor, and that shuts off the gene. But in the absence of tetracycline, so when you take tetracycline away, the gene now turns on. In this scenario, the RIDL scenario, the gene that turns on is a killing gene which kills the larvae. Now this is even more complicated because this is all based on the chimeric protein, and so there's engineered systems that are TET on and TET off, which means there are systems that exist that do the exact opposite of what I just said. Okay, so it's a way to turn on and off the gene by adding or subtracting tetracycline. So for RIDL, what they have is, so Oxitec in their factories will rear mosquitoes plus tetracycline in the water. Okay, the tetracycline will then bind to the TTA gene, which will bind here, and it will keep the killing gene off. Okay, then they rear a bunch of these males, they grow them up, they release them. Okay, so then you have the, we'll call these the RIDL males. RIDL males. They release them into the wild, they mate with the females, and the offspring of this cross inherit this killing gene. So they inherit the gene. So they inherit the toxin. But there's no longer tetracycline in the water where they're laid in the pond. So now the gene activates and it kills all the offspring. It kills actually larvae. Does that make sense? Do we understand how that works? Now you understand why I taught you promoters and why I taught you regulation of genes and all that stuff, because all that stuff is important for how you control genetic mechanisms to kill insects. Okay? Make sense? Fourth thing you need for a sterile insect technique to work is a way to release the insects. So Google is driving around in the vans, pumping out the mosquitoes, because mosquitoes can't fly like for miles and miles and miles. Mosquitoes basically come out, mate, and lay their eggs all within like the same neighborhood. So if you want to kill them over a large area, you need a way to release them. Same thing with the screwworm. So in the screwworm, they first tested this on an island north of Venezuela called Curacao. Okay? And Curacao was infested with screwworm, and they tested to see if they could eradicate the screwworm on this island. Islands are always the first testing site because there's no emigration in of insects. So what they did is they would drop the pupae out of an airplane, okay? So they just take the pupae and then they drop them out of an airplane as it's flying around, and that scatters them, and then they come out and they mate and produce a sterile offspring. And they did this for 6 months. So oftentimes with sterile insect technique, what you see is not only do you need a way to release them, you needed to release them like every day or every week, continuously for months and months and months. So you're just continuously releasing them. So they would release 2 times a week by flying around in an airplane and dropping pupae. And then it worked. They eradicated it in Curacao. So then the USDA said, well, let's try to get this out of Florida. So then they started in 1959 to do these releases in Florida, and it was working. And as I said, they kept pushing it. They kept pushing it from Florida all the way back, all the way down, all the way down, all the way down. And by 1995, it had already been pushed down to Panama. So we completely eradicated the screwworm from North America by 1995. And this is you'll hear about this a lot in entomology because this is the classic example of like awesome IPM that was able to completely eradicate a pest. And it's ecological, it's entomological, it's got a lot of cool aspects to the story. Not only that, but it's been replicated. So there was a shipment in the 1990s of contaminated livestock that was contaminated with screwworm that arrived in Libya. And then Libya had a massive outbreak of screwworm, and they replicated the techniques that the USDA used, and they were able to successfully eradicate screwworm in Libya. So it has like actually a second replicate of it working. So things you also need to consider. You need to measure, as you're sterilizing the males, you need to figure out a way to measure the fitness to make sure that their fitness is not reduced by the method of sterilization. So there's various ways to measure fitness, like abundance of the insect, size of the insect, fecundity. You can, in Drosophila, you can measure like how fast they'll climb up the walls of the tube. So behavioral assays will measure fitness. There's a bunch of different ways. But you have to figure out a way to measure the fitness before and after they've been treated with either the irradiation or the chemical sterilization to verify that they're not sickly. So that's always a big part. Some insects, so this is also important. So a lot of behavioral reproduction in insects is complicated, right? So Drosophila have special courtship rituals. I don't know if screwworm does, but a lot of the insects will have like behaviors that allow the female to know if this is a healthy male or not, okay? So a lot of times these courtship rituals have to be measured so that you know that the male can act normal after it's been sterilized. So there's a thing called mate discrimination, which we should talk about. So if you're releasing, so in the case of RIDL, where they're releasing RIDL males in South America, in Brazil, okay, the females are learning how to tell what's the RIDL male, or the wild type male, okay? They want to mate with the wild type one because this one will allow them to reproduce. So there's going to be huge selection for the female to figure out a way to tell the difference between this one and this one, right? Does that make sense? Because if she can figure out behaviorally how to tell the difference between this and that, then she's going to survive, and then her offspring will inherit that behavior to discriminate. So this is called mate discrimination. So the females are learning how to do that. And one of the reasons they do that is RIDL. So this is Aedes aegypti. Okay, and they made the RIDL with a research strain called Rockefeller. So this is a strain of Aedes aegypti that they had to build the genetic system into, right? This strain is going to be different from the local strains in Brazil. Okay? Because this has been reared in a lab for 50 years, right? So if you just straight up release these females, there's going to be a whole bunch of SNPs. It's going to be the same species, but there's going to be a whole bunch of SNP differences. And the females are going to be able to learn to distinguish, because some of these SNPs are going to cause behavioral differences. Some of them are maybe going to cause chemical differences in the smells, the pheromones. There's going to be a lot of stuff we don't know about. And the females are going to learn to tell the difference. So this is called mate discrimination. So you need to take steps to prevent this from happening. So the Rockefeller one is a strain from New York, and they're releasing this New York research strain into Brazil. So the way that you counteract this is back crossing. Okay, so we'll talk a little bit about back crossing. And this is a common research theme if you study Drosophila, even if you study yeast, if you study animals, anything, you talk about back crossing. So back crossing is if you have two different strains, let's say RIDL strain, Rockefeller, and you have local Aedes aegypti strain, And you want to get this one to be basically that one, okay? All you need to do is you need to take this male and one of these females from the local strain, and you cross them. And when you cross them, because insects are diploid and they are sexual organisms, their chromosomes will then, what happens to their chromosomes?
Cross.
Yes? What's that called?
Recombination.
Yes, we talked about this. They will recombine. Recombine. So the offspring, the F1s, if this is F0 cross, the F1s will be actually a mix of 50% RIDL, Rockefeller, and 50% Brazil. Okay? And then the next thing you do is you take this 50-50 mix, take a male, and you cross it again to the wild-type Brazil female, the local one. You do it again. Okay? And then this time, the F2s are 25% RIDL from recombination. 25% RIDL and 75% Brazil. Okay? And then you do this and you do it again. Again and again and again and again and again. And then eventually, the RIDL strain that you're going to use are now 99.99% Brazil. Does that make sense? So this is called back crossing. And you'll see this in yeast, you see this in behavioral studies. When you have worries that like a strain could have a different SNP that would cause it to behave different, then you use a back crossing strategy. The other way that we can combat mate discrimination is if we know how to make the transgenic insert, we can literally just take the wild population from Brazil and then do the microinjections again in that population directly, and then make a transgenic insect, RIDL transgenic, that is in that wild type population. But this is the easier strategy, the back crossing, because all you need to do is just set up a cage and they mate and they do it naturally.
And let me just emphasize, the reason I'm talking about this and the reason you would set up a back crossing scheme is because you don't want to be releasing New York mosquitoes into Brazil without knowing what kind of effects releasing those genotypes is going to have on that local population.
So that's a back crossing scheme, and it utilizes recombination to make two different strains the same thing. Okay, there's also new research into using drones for the sterile insect technique. So think about tsetse flies. Okay? So let's say we want to do sterile insect technique with tsetse flies. Tsetse flies live in the jungle. Okay? You can't have a van drive through the jungle and release tsetse flies. Maybe you could drop them by a plane, but maybe that doesn't work well. So maybe what you can do is you can engineer a drone. Bad drone. Okay, you can engineer a drone to go around and to go down close to the ground and then drop the pupae in different areas. Or in mosquitoes. So let's say you're in Alabama. And you got different counties. You're doing your sentinel chickens. And you see West Nile virus is like way intense right here. But in here is like the forest in the boonies. And you don't want to go drive out there. So maybe you have a drone, take some RIDL eggs, take that drone, you have them drop them in the ponds in this intense forest area, okay? Then it's a way for you to like target where you're doing the sterile insect technique, okay? So there's a lot of like new adaptations. So people at Harvard are working on developing drone-mediated dispersal of SIT. Also, Omar Akbari has a paper on this.
There's also someone here that's like
the nematode person.
Yeah. That's not as
He's kind of like using drones to look at like agricultural fields, and then you can take a picture, and then you can like measure by like the greenness whether or not there's like a disease is floating around and killing the plants. But yeah, similar thing. Like there's a lot of people trying to figure out ways to use drones. So advantages of sterile insect technique. Why is this so big? So if you think about it, the only other way we have to control mosquitoes is insecticides, pretty much. So you put insecticides in a pond, and insecticide is basically going to kill every insect in that pond. You're not just killing mosquitoes, you're killing every insect in that pond. So that would be what are called off-target effects, okay? And there's lots of beneficial insects like honeybees. So a lot of honeybees get their colonies killed because they go around and they pick up pollen that is contaminated with insecticides. Okay, and they bring it back and then their colony dies. So this is a huge issue, is how do we limit the off-target effects of the insecticides, because they're basically not specific. Okay, but sterile insect technique is always, by definition, species specific, because it only works if there's a mating event. So if you release a bunch of screwworms that are sterile, they're like it's like a homing mechanism. They are going to find the females, and they're only going to mate with those screwworm females. And so the only reduction, the only killing you're going to get is species specific. So there's very, very low off-target effects. It's local. So like I said, one of the main things you need a way to disperse them. That is good and bad. It's bad in that to get it to work, it takes a lot of energy and a lot of input. But that also means like you're not going to have massive global effects on ecological populations in southern United States if you're only releasing sterile insects in southern Florida, right? Like it's only going to be in southern Florida. So it's local. It's not going to spread. It's species specific, and the insect is like a homing beacon. So I'll finish with this. There's a lot of stuff that the USDA does that you never hear about. That like it's really cool, but you just never hear about it. So in California, I guess California is something like that. So California has lots of ports, right? And it's always getting like imports of goods from China,
Coronavirus.
from Russia, like stuff's being imported into ports of California. Okay? And one of the huge concerns is California is a massive industry for fruits. Okay, so in California we grow blueberries, strawberries, all that stuff is grown in California. And with all these shipments, we worry about importing fruit flies. Okay, not Drosophila fruit flies, but like the bad fruit flies, Tephritids, which look like that. They're fruit flies that actually like cause massive economic damage to like raspberries, blueberries, berry crops. Okay, so there is a team at the USDA that if they monitor in California, and if what they find, and it actually happens quite a bit, is oftentimes they'll find a little explosion of imported Tephritid.
And we know where they're coming from because of their genetics.
So if they get like a Tephritid from Asia that comes here and it starts establishing itself and it starts spreading, there's actually a team at the USDA which will make sterile Tephridids, and they'll come to this specific spot and they'll do sterile insect technique in this local region to push back the expansion of the imported population until it's gone. So there's actually like a response team, and this is being measured so that when this happens, if we get an imported pest, the USDA will send a team to do the sterile insect technique there. So this is just one of the really cool things you don't really know is happening. But they do that.